second. All right. Uh-oh, don't do that. Share screen. This is where we left off. Can everybody hear me? I've been having issues kind of with my internet today. All right, can we all see this? And you can hear me? Thumbs up? Okay, great. We've been talking about viruses and the different structure of viruses. And now I want to get into their modes of multiplication, because that's really what they're all about, right? They just want to multiply. It's a downstream consequence that it causes infection and makes us sick and can give us cancer, can kill us. Their whole goal, though, is just to persist, right? And so there are these various phases that are, pretty similar to the steps that we talked about earlier of how microbes mount infection. But we break them up into these categories. Absorption, so that's when the virus has to bind to the host cell. Penetration, the genome of that virus has to then enter the cell. Uncoating, so once it gets inside the cell, that capsid has to break apart and release the genetic material. Synthesis, it then needs to either go to the nucleus, if it's a DNA virus, or set up shop somewhere specifically in the cytoplasm, if it's an RNA virus, and then start copying its genome, start replicating the genome, start translating the genome into proteins, and then reassemble those newly minted pieces into a nascent viral particle, and then finally release. And there are two major ways. It can either bud off of the cell, which keeps the cell intact, or it can lyse the cell, break the cell open, which clearly will kill the cell and not. So there are two major ways to exit. And here's an illustration looking at that. So for step one, you have absorption, and this is going to be the cell attaching, I'm sorry, not the cell, the virus attaching to the cell using the spikes and then binding to corresponding receptors. So again, with SARS-CoV-2, it's got spikes, and those specifically bind to the ACE2 receptor. Once it binds, it then needs to enter. And this is often done by the cell, triggering the cell to just engulf it, okay, into a vesicle. So this, it binds to the receptors, and then it sends a message to the actin filaments, and then those actin filaments polymerize, and then it swallows it in. Then it uncoats. It takes off that vesicle on the outside. It takes off, if it's an envelope, it's going to lose the envelope. And then it has this capsid here. And the capsid is going to start breaking apart. And once the capsid breaks apart, then we have synthesis. We have the RNA, in this case, because it's in the cytoplasm. Not all DNA is in the nucleus. Not all DNA viruses replicate in the nucleus, but most do generally. So the RNA is going to go, and it's going to have the replicase that copies a bunch of new RNA strands. And then it's also going to have translation. It's going to use the host's machinery, the ribosomes, to translate new proteins, like for the capsomers, for the capsid, the spikes, etc. And then all these pieces are going to make their way to the inside of the membrane again, where they assemble into new particles and then release from that host cell. And that's the major steps of viral infection in a nutshell. Okay. Now, viruses don't have motility. They don't have flagella. They don't have sensors they don't do chemotaxis, right? They very coincidentally come in proximity with a susceptible host cell. Okay, it's not by them targeting that host, it's by the behavior of the host. Okay, and when that happens, it's somewhat of a serendipitous event. And these viruses have to have a very specific host. It's species specific for the most part. It's tissue specific for the most part. Hepatitis B is going to infect liver cells. Poliovirus is going to infect the intestine and then migrate into the nerve cells. Rabies is one of those that has a broader host range. It can infect many different mammals, and it can infect various different cells. Okay. But for the most part, generally speaking, it's a narrow range at which a virus can set up and infect. Right? They have to get in the cell. Viruses cannot replicate outside of a cell. It cannot happen. So they have to get inside the cell to use the components that the cell itself uses to replicate its genome and co-opt it to replicate the viral genome. And as I said, they enter through fusion if they're envelope viruses. And so that means that they bind to the receptors and then it just kind of swallows it in. Or they can use a process called endocytosis in enveloped or naked viruses. And endocytosis is a cellular process that is, if you've had cell biology, you've learned about this, is it's a way to engulf other things that it's supposed to internalize. But the virus has hijacked that mechanism and used it to then engulf the virus. Okay. And then there are kind of two different pathways depending on if you're the RNA flavor or the DNA flavor. But essentially, you're going to bind to the cell. You're going to be internalized, in this case, engulfed. In this case, at the top, in the top example, what you're looking at is the virus binding and then really just pushing the capsid inside the cell. So it's not enveloped, the whole virus itself isn't enveloped into the inside of the cell, the host cell. It just binds and then pushes in its capsid containing the genome, and then that uncoats. Whereas below, you create this endocytosis process, which engulfs it. You have now surrounded it with some of that membrane on the outside host cell, and then that will start to break down and release the genome, which will then go typically to the nucleus. This is a naked virus, so same kind of thing. It binds to the receptors, the cognate receptors, and then it internalizes it in the form of a vesicle, and the vesicle breaks down. Okay, so slightly different things to achieve the same overall goal of being transported inside that host cell. The replication and the production of proteins depends on the type of virus, DNA or RNA, as I've said. Generally, DNA viruses replicate and assemble in the nucleus or near the nucleus. RNA viruses typically replicate and assemble in the cytoplasm. There are some DNA viruses that will replicate in the cytoplasm as well. There are DNA viruses that will replicate in the nucleus and then send the RNA out into the cytoplasm to then be translated into the proteins and stuff and then assemble out there. But generally, that is the domain that these different viruses use, either inside the nucleus or outside the nucleus. And it's just simply because that's the nature of the host and how it uses RNA and DNA, right? DNA of the host is going to be transcribed in the nucleus, and then the RNA of that is going to then leave the nucleus into the cytoplasm to be translated into protein. So the virus is just really, again, using these natural processes that the host cell itself uses in order to benefit it, okay? And we said before yesterday, you can have positive sense RNA or negative sense. Positive sense is ready for translation. Negative sense has to be converted into positive sense before it can be translated into proteins. Once that translation happens and that transcription happens, now the virus has to assemble into a new particle. And this can happen in different ways. But first, you really need the capsid. As all viruses, we said, have a capsid. You need to start forming this capsid. The capsomeres assemble, and then they are there. It hasn't closed up yet. It's got to then get that genome in it, whether it's a DNA genome or an RNA genome. And oftentimes, what you'll see is that it will cause tissue that's been infected to kind of merge together and get this giant cell with multiple nuclei in it, which is very, right here, which is very distinguishable from just a normal cell with one nucleus. And that's how pathologists can diagnose certain viral infections by taking a biopsy and looking at the tissue. This was done, of course, before it was so easy to just do a PCR test and sequence the genome. Okay, release, also called egress, so leaving that cell, budding or exocytosis, or lysis, right? Those are the three major ways. And they have their benefits and their disadvantages, right? If you lyse the cell, you kill that cell, and that can signal to the host that something's wrong. Whereas if you bud out of the cell, you may not, you may go under the radar, so to speak, which is much, more advantageous to the virus that wants to be stealth. Also, exploding the cell can lead to enough tissue damage that that host dies. And if the virus doesn't have an opportunity to get outside of the body, it doesn't just need to get outside the cell, it then needs to get outside the body. If it can't do that before it kills the host, then its legacy dies with the host, right? And so this is how the assembly and release occurs. You have here's where my draw, here we go. So we have the capsomere proteins and the genome both moving together and packaging it up. Anybody know? So if the capsid looks like this, what kind of capsid is that? There are two types, right? What kind of capsid is that one? Helical or icosahedral?
Helical, I think.
Helical, right? The icosahedral has that more geometric shape. A helical capsid is going to be more tubular, all right? That capsid then has the genome packaged, and also other proteins that have been translated are sent out to the cell membrane. So the viral matrix proteins and the spikes, those things embed themselves into the host membrane. The capsid then aligns itself to that viral matrix protein. And once that happens, it stimulates the budding or the exocytosis of an enveloped virus, such as this, okay? And now it buds off, and you have a free infectious particle that has taken with it some of the host membrane. Which then what? Does that make that more susceptible to a shorter life or less susceptible to a shorter life? Or that's kind of an awkward way of putting it. Do envelope viruses, are they going to live longer than naked viruses?
No, shorter.
No, they're not going to live as long because they have that host membrane that doesn't like to be outside the body. What are some of the effects of having viruses making babies in your cells? Well, we call these cytopathic effects. Cyto meaning cell, pathic, right, meaning to damage, not normal. Abnormal cytological effects. So we can look at that with the microscope. As I said, you can see it transforming or disorienting these cells, okay, in their shape. It can also make them form inclusion bodies or a syncytium, where a bunch of cells now merge together, and that's how you get this multi-nuclear cell, okay? These cells are binding together. Or sometimes viruses can make new particles, and then those new viral particles exit the cell, but then go into the neighboring cell, right? And then that's how they start perpetuating the infection. And that forms this syncytium, this kind of long connection of cells that are now almost like, you know, like, well, I don't want to say that. I want to say that. Kind of, well, yeah, no, I won't. That's, no, never mind. The point is that these cells all get together and they're now connected and allowing the viral particles to pass from one to the next. Persistent. So those are going to be infections that go into a latent state, a chronic infection. So it's going to be there with the host for a long period of time. Measles, for instance, can have the initial outbreak and the initial effects, but then remain in a latent state in the brain for years. Herpes, which we'll talk about when we get into DNA viruses, they cause things like cold sores if it's herpes simplex 1, or genital herpes if it's herpes simplex 2. And those things don't go away, right? The herpes virus that causes chickenpox. If you get chickenpox, you can later on in life have that remanifest in the form of shingles. If your immune system is experiencing a stress, or you're experiencing a stress. So some things don't just pass, some things stay around. And these things, I mean viruses. Some viral damage can result in cancer, right? So the viruses that insert their genome into your genome are going to disrupt your genome as a result. And so when you are translating that gene into a protein, but it has now another piece of DNA in it, that will change the protein that it makes. It will either shorten the protein or make the protein longer, or give the protein a different shape, which then renders its function useless because proteins are all about their conformation, right? So if you change one little twist or bend in a protein, that may not be able to further its function. And if that site where that virus inserted its genome happens to be in an area that dictates how cells divide and when to stop dividing, those cell cycle checkpoints that we talked about when we were discussing HeLa cells, then that can lead to cancer. The cell has effectively been transformed and it is now oncogenic. It creates cancer cells. Okay. Viruses that do this are called oncoviruses. Onco is cancer, cancer viruses. So papillomavirus causes cervical cancer. It's very likely. I don't know if Henrietta Lacks had papilloma or not. The Epstein-Barr virus can cause Burkitt's lymphoma. There's a JC virus that can cause cancer as well. Okay. And so that, of course, is a long-term issue caused by viruses, right? We talked a little bit about bacteriophages, right? And now they're, you know, it's even more relevant because I just mentioned the announcement of the CRISPR that was awarded to two female researchers, the Nobel Prize in Chemistry. If you guys didn't catch that, if you signed on a little bit later, is exciting, right? And so these bacteriophages are viruses that specifically infect bacteria. It's going to have a similar process of getting into that bacterial cell as do viral entry into animal cells. But there's no nucleus really per se in a bacteria, right? Because a bacteria is a prokaryote. And how are prokaryotes different from eukaryotes? Somebody? What are they missing?
No nucleus.
No nucleus. Yes. No other membrane-bound organelles as well. So it's a little bit different in terms of their replication. But they all release via a lytic cycle. They disrupt the bacteria, rendering them dead, which is why some bacteriophage have been used, as I mentioned, as a therapy to treat humans that have bacterial infections that are resistant to the antibiotics that we have been using. So the idea is, well, let's find a bacteriophage, a virus that's harmless to the humans, but detrimental to that bacteria. Briefly, let's talk about prions. So prions aren't viruses, but like viruses, they aren't alive. They're even more simplistic than viruses, though, because they're just protein. There's no genetic material, okay? There's no co-opting of the host's machinery. All it is a misfolded protein. And remember what I said, if you change the conformation of a protein, you render it useless. And so what these prions do is they infect tissue, and their misfolded protein shape influences the structure of other proteins. And then you get these aggregates of misfolded protein that have no function and are just taking up space. And we see this as disease mostly in animals, in our livestock. And it's always cerebral, right? So encephalopathies, mad cow disease. If you look at a section of their brain, you'll just see all these holes like Swiss cheese, and they just end up going crazy because their brain is literally being reconfigured and it can no longer work. In humans, there is Creutzfeldt-Jakob syndrome, which is caused by proteins. Yes, what? Sorry. No. Um, and so this can be an issue with humans as well, but typically um, it's a bad thing for our livestock. And this is just showing that we have these misfolded proteins, and they're depicted by these little bends here, and then they get into proximity with these correctly folded proteins, and then they influence that structure of theirs onto the correctly folded proteins, and you get these aggregates of a bunch of misfolded proteins that then will attack the cell.
Hey, Carl.
Yeah.
I have a question. So I thought mad cow disease was caused from it's kind of gross, but like people taking cows and then repurposing them into more cow feed and just feeding cows meat, essentially.
Well, yeah. I mean that is a way to introduce. So if you're feeding a cow that had prions, then that's the delivery system.
Okay, so it's not just the act of like causing an animal that doesn't eat meat or like to be carnivorous. Technically, it's just the it's infecting another cow with a cow that's already been susceptible to it.
Yeah, most likely. Or I mean, I don't know. Perhaps, um,
Because I thought it had to do with the whole cannibal factor of it, because like you're not, but I mean that could have also that's just what I was told when I was younger, so I don't know.
Yeah, no, I don't think that it has to be cannibalism to that, but that might be a really great way, not great in terms of fantastic, but like a very efficient way to spread the prions from animal to animal. And it's possible that the generation of prions might happen when an animal consumes its own protein from the same species, but I don't know if that's the case. And then finally, satellite viruses and viroid. Satellite viruses cannot infect a host on their own. They need to tag along with other viruses in order to do that. So adeno-associated virus, that will only be able to replicate in cells that are already infected with adenovirus. Okay. Viroids are going to be short pieces of RNA, but no protein coat. Okay, so kind of like the opposite of prions. Instead of being just protein, these are just RNA. And so far, I think we've only found things of this nature in plants. Typically, RNA is very, very unstable and will break down if it encounters temperature differences. There's all kinds of enzymes just in the environment that break these things down. Okay, so that is the end of that chapter, chapter 6. Any questions on that before we start going into specifically DNA viruses?
I have a question about the CRISPR thing that you're talking about.
Mm-hmm. CRISPR.
That's okay right now. It's like the new discovery that they came up with, like in the last year, like of how to do it like more like with just inserting like one in specific nucleotide, or is it like the actual invention of CRISPR that's been around for a longer time?
So the adaptation of CRISPR as a genome editing tool came out, I believe, in 2012. Because when I was still in grad school, we were using it in our C. elegans virus model to change the genome to see if certain genes increase susceptibility to infection or decrease susceptibility. So, and I finished grad school in 2014. So yeah, it had been around. It was new during that time. Mm-hmm. But the idea, the discovery of CRISPR goes back to I think the 70s. Okay, it was identified as so CRISPR. What does it stand for? It's CRISPR.
Cas9 repeat inspector palindromic repeating.
Yeah, that's close. So like repeating, inducible, palindromic, yeah, repeats.
Telomere.
But anyway, so what it is, it's a bunch of similar segments of genomic sequence that repeats over and over again. And so they found this as they were getting sequencing techniques improved. They were looking at bacterial genomes because they were small. They were the easiest things to sequence when at a time when you couldn't sequence a whole a human genome. And they found these repeating units. And so that alerted them to some interesting thing, but they didn't know what it was used for. And then slowly scientists started finding out the link between between the Cas9 system and its use to basically prevent infection from viruses. But so it's a long piggybacking story. It wasn't until 2012 though that researchers were able to adapt it and put it just those enzymes together with an RNA guiding strand to then use it to manipulate genomes. Does that answer your question?
Well, kind of, but so is it that whole thing that won the Nobel Prize, or is because I know like the last year they made an advancement where they can get more specific on it too?
Yeah. No, it was yeah. So since 2012, researchers have been perfecting it more because, as you said, there are a lot of off-target sequences that the CRISPR can attach to because it's a very short piece of RNA. And so the specificity leaves something to be desired, which means if you want to target a specific gene, you can do that, but it might also target some other genes that are similar enough in their sequence, and then you get downstream effects from that. And so, yes, they've been improving it, getting it more specific. This Nobel Prize is to the two women who basically took CRISPR and made it a tool for genome editing, not for the, downstream perfecting of it.
Yeah. Okay. Thank you.
Any other questions before we get into it? All right, we have about 10 minutes, so we won't get through DNA viruses, but we can at least start into them and give you an idea. Okay, there we go. There we go. All right, so in the context of viruses that are infectious to humans, which is what I want to focus on, the DNA viruses are going to be double-stranded. And as I said on Monday, most DNA viruses are double-stranded. There are exceptions, though, to everything. And parvovirus, for one, is an example of a single-stranded DNA virus. RNA viruses are going to be basically single-strand, except for, of course, the double-stranded reovirus. So there are some exceptions to the rule, but again, most DNA viruses are double-strand, 2 strands of DNA. Most RNA viruses are single strand. And then the RNA, yeah.
This might be a silly question, but so the ssDNA parvovirus, is that just single stranded? So it kind of tells you the beginning of the virus's name if it doesn't have a normal strand like composure. This is ssDNA parvovirus. Is that like a commonly written thing? Because I saw that on some of the other slides, and I'm just wondering about that.
The ssDNA, that just means single stranded.
Okay.
Yeah. And if you see DS, it's going to be double-stranded. Just like ssRNA is single-stranded, DS is going to be double-strand. So just a shorthand. Here's a table of some common DNA viral pathogens. Again, you don't have to memorize these tables, but this gives you a nice overview of the type of virus, and then where it infects. And so you can see we have different types that infect different areas, whether it's the skin or the salivary glands or, the eyes. We'll talk about keratitis. There's a herpes virus that infects your eyes. So they have a bunch of different locations. And this is just to give you a general overview. You don't need to memorize these tables, okay? Most DNA viruses, as I said, multiply in the nucleus. Most RNA are in the cytoplasm, but again, there's exceptions to that as well. Some can be asymptomatic, some can be mild, some can be life-threatening, and that can all be one type of virus doing that, as we well know right now with COVID. SARS-CoV-2 can be mild in some patients, can be asymptomatic in other patients, and can unfortunately kill other patients, okay? So it really just depends. It's not the type of virus that manifests in these different ways. It's a single type of virus, and it just depends on the patient has the misfortune of coming into contact with it. Some viruses are strictly human in origin, so it means you can only get it from humans, like HIV. Others are zoonotically transmitted. So remember, zoonotic or zoonoses is infectious disease from animal vectors. Okay, so think of West Nile virus or dengue virus. Those are viruses that come from specifically, arthropods, mosquitoes, okay? Typically, when you get a viral infection, it's going to be a rash. You'll get a fever because your immune system is starting to ramp up and detect this infection. You might have muscle aches, feel like you're really sore, and that can be from the cytokine storm that is being released that we talked about. If it's a respiratory illness, you'll have difficulty breathing. And as a result, your body is going to start producing interferon. Remember, we talked about that, how that's the cell's really first way to warn other cells about a virus that's infected it. And then you get, of course, your cytotoxic T cells to take them out. You get the production of antibodies from the B cells. And hopefully you have immunity afterward. Viruses can be lytic in nature. And that's going to be a very quick course of infection. If it's in the lytic cycle, where it's, breaking open the cell and releasing itself. Some, however, can be latent infection, where they don't release themselves and then shed into your gut or out of your respiratory tract. And those latent infections are going to come back and haunt that host later on. Or, you can have a chronic infection where it's detectable in the tissue, but it's really dividing and making new viruses at a super slow rate. And it's so slow that you really don't have any symptoms, or you might have very mild symptoms, okay? So those are the three types of infections: lytic, quick, think of things like the flu, that's a lytic cycle. You can have chronic infections, think about things like herpes, or you can have latent infection, things like varicella, which is chicken pox, and then it goes and hides out for a long time and comes back as shingles. Okay. These persistent viruses, why are they persistent? Well, they're persistent and they're chronic because they have inserted their viral genome into your cell, like HIV. So you can't get rid of it now, because anytime your cell thinks it's doing its job and transcribing its genome, it's ripping off that viral genome and shooting it out there into the cell to then perpetuate. Or it can be oncogenic, as I said, it can cause cancer. Some viruses cannot cross the placenta. And so if you have a mother who has a certain viral infection, it won't be passed on to the baby in utero. It might during the birthing process, which is why sometimes they provide C-sections when mothers have known outbreak of a certain viral infection. Some can, though, cross that placenta and infect the baby, and there can be sometimes permanent defects, teratogenic defects. Think about Zika, right? That can cause microcephaly in the babies. It makes their heads not normally sized as a result of the virus crossing the placenta. Okay, I think this is a good place to stop. We will then pick up Friday, wishful thinking. We'll pick up Friday with the different types of DNA viruses and go into them and talk about them a little bit more specifically, okay? So you have 10 minutes until the quiz if you want to take it again. If you're happy with your score, then by all means, don't need to. I set it up for a second attempt. If for whatever reason it's not working, shoot me a message really quickly and we'll see if we can make that work. What else? There'll be a quiz Friday, right? I don't think it will be, there will be some immunology stuff, but it'll be immunology and virus related stuff on the quiz on Friday. Immunology wise, big picture stuff. What's a B cell do? What's a T cell do? That kind of a thing, okay? Any other questions? If you want to sign off now and review your material for the quiz that you want to retake, by all means, go for it. If you want to hang out and ask some questions before or after the quiz time, I'm here. So any questions?
I had a quick question about the prions. I think I learned before that they are what cause Alzheimer's. Is that right?
No, but it's a similar mechanism. So with Alzheimer's, you have beta plaques and tau tangles, and they are proteins that aggregate and stick together, so you have a bunch of different proteins that are now globbed together, which then interfere with normal cellular processes. And so it's a similar mechanism as prions, but it's not a prion per se.
Okay. Thank you.
Mm-hmm. Prions disease is not that common in humans as far as I know. Alzheimer's and other dementias are fairly common. And I don't know all the science behind that, but we're really starting to get a better idea of what makes these proteins aggregate like that later on in life. And coming up with new ways to detect it, right? So the problem is once someone has these tau tangles and these beta plaques, at that point, it's really hard to intervene and do something to correct it, right? So ideally, you want to be able to detect the production and formation of those proteins before they can then start to wreak havoc later on, right? And so what's the screening process, right? I mean, if you have a family history, then you should go get tested. But I think they're still really kind of trying to work that out.
So I had a quick question. So the quiz Friday will be mostly chapters 6 and 24?
No, 24 is DNA viruses, right?
Okay.
The quiz on Friday will be some big picture immunology stuff.
Okay.
And then some of the stuff we discussed Monday and today, chapter 6. So just big picture virus stuff, right?
Okay.
That help?
Yeah. Thank you.
Hey, Carl.
Yes.
I was supposed to get my work schedule changed permanently around, but they're not going to start it till next week. So could I maybe take the quiz at before 10 or after 4:30 on Friday?
Yeah.
Early or late, whichever one you prefer.
Let's do early. Let's do before 10. Let's do it 9 o'clock. 9 o'clock work? Okay.
What?
9 o'clock, does that work?
Yeah, I thought you said 8, and I was like, oh, okay.
5:30. I'm up by then, thanks to my young children. So
Yeah, I could do it, but I'm just not usually awake at 5:30 if I can help it. Yeah, so 9 works perfectly for me.
no, I don't. I strongly believe that you shouldn't have to use your brain before 9 o'clock in the morning. I had an organic chemistry class that started at 8.
I
And
have that right now.
Sean starts at 8?
Yeah, Sean starts at 8.
Oh, ouch. Yeah, that's rough. I don't like to have to, especially back then. I did not like to think until 9 o'clock. So I won't do that.
Yeah, perfect. Okay, thank you. Because I feel like and it's that early. I spell until like 7:50. I'm just trying to just like chug caffeine. So it's
Yeah.
not fun.
Yeah, no, I get it. I get it. Get back up to baseline.
Yeah. All right. Well,
Oh, you're muted.
so I will see you on, I guess, next week, and then also science club meeting at 1 on Monday. Is that what time we should shoot for?
Yeah, let's do that.
Okay.
So
I will-
send out an email to the group. I need to send out an email too with the link to our presentation event yesterday. So, but I'll let you take care of notifying them about the next meeting.
Perfect. Okay. And what do you think about trying to do like discussions where people try and like bring up like ideas of like different scientists and stuff, or like people that didn't like unknown, underappreciated, maybe?
Absolutely. I
And
think
like discussion like topics or like forums like on that.
I think that would be great. I mean, I leave that up to you guys, but there's a whole lot of science out there that gets credit and a whole lot that doesn't, and which is why I was wanting to shine a spotlight on HeLa cells and on this Tuskegee project. It doesn't have to be anything that's controversial, but yeah, I mean, it could be. There's several that I have in mind that are, and I can pass those on, or you guys can come up. Obviously, I want you to come up with your own things.
Yeah.
To
I think I might
for you, but
I might shoot out that idea topic like in the email and be like, if you guys have anything you want to talk about or just like share with the group, like
yeah.
Okay.
Yeah.
Perfect. Okay. Well, thank you. I will talk to you soon.
Okay. Bye.
Hey, Emily.
Oh.
Oh shoot, it just froze.
She's gone.
Did we lose her?
She
Darn it.
signed off.
I'll email her. I started listening to some different podcasts that seem really awesome that have to do with microbiology. And some of them are just like a couple hours long of their bunch of people sitting around, kind of like journal clubbing. And then others are a little more specific, but I wondered about maybe some of those for topics.
Absolutely. Yeah. I think, there's obviously business stuff that the science club needs to do, but it should primarily, it should be fun, right? We should be able to talk shop and get into it like that. So absolutely. I think that's a great idea.
Are there any do you do podcasts? Do you listen to podcasts? Are there any that you highly recommend?
From a science perspective, I like TWIV, but so TWIV, TWIV, it's This Week in Virology. So what
I got that one.
Oh, did you? Yeah.
I got that one. Mm-hmm.
Yeah.
Yep.
I like that one. I know that, was it Rachel that sent an email with some of the podcasts she was listening to? Someone sent an email out with a list of podcasts, and let me look at that one. I do TWIV, and that's really occasionally I'll do the Seth Rogen podcast because he'll bring in certain scientific topics. But,
really? Okay. Now TWIV, that's because I guess what I had is TWIM, So
Oh,
if there's
this week
another
in
one,
micro. Yeah, so TWIM is this week in microbiology. TWIV is this week specifically in virology. So same kind of concept though. They talk
Okay.
about, oftentimes they'll have an exciting paper that comes out that week, and then they'll discuss it. And it's nice because you get these really very very profound thinkers that are experts in their field discussing scientific papers, and it's a good way to see, to learn tricks and techniques and ways to evaluate that are sometimes over our heads until we do it a lot. So
awesome. Thank you. Yeah, I know the TWIM one. I think they're like that. It kind of seems like the same thing. They're some hours long and they're really getting into it.
yeah,
It's awesome. I listen to them while I house clean.
Yeah.
That's my job, and that doesn't take thought. So
nope, that's the time.
okay, I'll look into the TWIV. Thank you.
Yeah, it's
All
good.
right.
I've often thought about using them alongside with the journal club. So pick a TWIV or a TWIM where they look at a paper and then have you guys read the paper and then but listen to that and then see how their analysis differs from your analysis or how similar they are. But
They'll be way more in depth.
as it should be at this point, right?
Yeah, it's pretty awesome. All right, thank you.
Bye. See you, Christine. All right, see you everyone. Have a good one, and I'll see you on Friday.